Afternoon, my dear. How are you? Efe, I can see you. I can see the real Ila. Which other person is there? Terelia, Efe, Uma, Sandra. I can see all of you. Yes, you are entering. So we thank God for today that we are taking off from where we stopped last time. Last time we looked at the last time we looked at the leaf. We studied the general structure of the leaf, the anatomy, the physiology. From there, we looked at pollination. From pollination, we looked at different categories of pollination. We also looked at how the diagram of fertilization process. We look at self and cross-pollination. We also look at characteristics of self-pollinated flowers, characteristics of cross-pollinated flowers. We looked at fruits and seeds. We discussed fruits. We discussed how seeds are formed, difference between dicot and monocot. And then we finally look at classification of fruits according to whether it is dehiscent or simple and simple fruits. And then we looked at the simple fruits, different types of simple fruits, dry dehiscent and dry indehiscent. And then we also looked at capsule, caryopsis, and then achene. Then we looked at nuts, Samara, Cypsela, and then we looked at fleshy fruits, the drupe, the berry, the aggregate fruits, and multiple fruits. That's what we discussed last time. So today we are going to enter into a very important topic and very, very significant for any person in biology. So we are going to look at genetics. We are going to look at genetics, Mendelian genetics. You see, genetics is a science of heredity and variation. It is a science of heredity and variation. And what is heredity? Heredity is the tendency of an organism to resemble the parents or siblings. The tendency of an organism to resemble the parents or the siblings, the young ones, is heredity. And then we also have variation. What is variation? The tendency of an organism to be different from the parents and preferably the young ones or the siblings. So these two, science of heredity and variation, is what we call genetics. And any living organism also exhibits characters that show that genetics is a reality. And then, who is the father of genetics? Gregor Mendel. Mendel was a graduate of physics and mathematics, or what we call graduate of combined physical sciences in his undergraduate days. But when he finished university, he has a call into spiritual ministry. So he now entered the monastery to become a priest. So he was a monk in the monastery. So something happened. His knowledge of basic biology and his knowledge of mathematics and physics now came into play because where he was housed in the monastery, there's a lot of flowers around that place. So what he did was to begin to play with these flowers and then begin to do pollination. He now do self-pollination. He brought white flowered plants and red flowered plants and then pollinated them, do the cross-pollination, hoping that what we get is 50% red and 50% white, that they should be equal in terms of the number that will come as offspring. But he was surprised to his bone marrow because the first generation were all red. Mendel was thrown aback. He was surprised. What has happened? I pollinated red flowered plant with a white flowered plant. I'm supposed to get 50-50, 50% red and 50% white. But here was Mendel that did not get that kind of result. He got everything 100% red. Then Mendel relaxed and said something must have happened. So what he did was to cross-pollinate the young ones. And then what he got was so surprising because he got 75% red and 25% white. So in the ratio of 3:1. So that was what Mendel did. Even though Mendel kept his work, but people never recognized Mendel. They never agree with him. His work was thrown into, nobody recognized his work. It was many years after his death that people stumbled into his work. Then people now begin to realize that, look, oh, this was what this man was doing. And so that was why Mendel's work now came into view. It was blown up. And then Mendel was now celebrated as the father of what? Genetics. So before we go into Mendelian work and all these things, we want to look at genetic terms that are familiar we get in genetics. Remember that genetics cut across all the biological sciences and even medical sciences. So in plants, you also have genetics. In animals, you also have genetics. In medical areas, you also have genetics. In microbiology, you have genetics. So this course cut across all disciplines, all the disciplines in life sciences. So let's look at some terms we encounter in genetic study or in the study of genetics. One is heredity. And what is heredity? Heredity is the tendency of what? The transmission of characters or traits from what? Organisms to their offspring. The tendency of transmission of traits from the what? From the parents to the what? Offsprings. I've already defined it. The ability of the young ones to resemble the parents or the siblings. That is heredity. Then variation is the tendency to be different. So sometimes one may not resemble the father. One may not resemble the mother. Then, or even the siblings. So that one is variation. So when organisms do not resemble, that one is variation. It doesn't mean that everything is 100% different. It could be some hidden characters that you may not see at that initial time. So we now look at what we call gene. What is a gene? A gene is a unit of inheritance. It is gene that transfers character. That's unit that transfers character from parents to what? The offspring is called what? The gene is the basic unit of inheritance for given characteristics. Now, you may not see the gene with your naked eye because you find out that the gene is a chemical substance that is found inside the chromosomes, which is found inside the what? The nucleolus, which is found inside the what? The nucleus, which is found inside the cytoplasm of a cell. So when you talk of gene, it's not something you can just hold and touch your hand. Something is microscopic. Something is in that lower level. That chemical substance, it exists because you do not resemble a goat. You do not resemble a chimpanzee somehow, but you resemble your parents. You do not even resemble people in your village, some other people in your village. You are peculiar character. A family is known, some families are known to resemble each other because of this word gene that transfer traits from parents to the offspring. Then you also have what we call the chromosomes. As I told you, that the gene is a chemical substance found inside the what? The chromosomes. In fact, genes are located on certain positions in the chromosomes. The position of a gene in a chromosome is called what? Is the position, a gene, because Genes are not scattered. They occupy certain position, and that's what we call locus. The position of a gene on a chromosome is called what? Locus. The plural is loci or loci. So these genes are housed by the chromosomes. So what is a chromosome? A chromosome is a rod-like substance that contains the word, the gene. And if these organisms have specific number of chromosomes, for example, if you pick the cell of a man, if you pick the cell, just a single cell of a man, check the number of chromosomes. The chromosomes are diploid number, depending whether it is picked from the body cells, with body cells like head, like hand, like leg, like eyes, like nose, mouth. These are the body cells, tongue. So when you pick a cell from the body cell and check the number of chromosomes for a human being, the number of chromosomes is 46. But these chromosomes are in pairs. That's what we call diploid number. So chromosomes are rod-like substance that contains the word, the genes. An organism has specific number of what? Chromosomes in them. For example, I talk of human beings. We have 46 number of chromosomes in our what? Body cells. But we have 23 number of chromosomes in our sex cells. Sex cells are the reproductive cells, like the sperm and the ovum. And these are the sex cells. So the number of chromosomes in these sex cells are 23. That means they are half the number of chromosomes in the body cells. So why the number of chromosomes in the body cells are 46? That is diploid number, or in bracket 2n. 2n is a kind of diploid number, complete number of chromosomes. But a sex cell has haploid number of chromosomes. That means they have half the number that's supposed to be present in the body cells. So that is a haploid number, or what in bracket n. So while diploid number is in bracket 2n, the haploid number is in bracket n. And haploid number is found only in the sex cells. That means the ovum or the sperm. And diploid number are found in the what? Body cells. Cells of the eye, cells of the nose, cells of the eye, of the ear, cells of the legs, cells of the bottom, cells of the legs. They are diploid number in bracket 2n. So that is what we call a chromosome. Now, we want to look at what we call an allele. Allele is a pair of genes that control a given character. If a gene that controls tall is represented by capital T, or capital T small t, because they may be homozygous or heterozygous. This gene, a pair of genes that controls a given character, is called what? An allele. And this allele can be identical, they can be similar, identical, or what we call homozygous. In that case, we have capital T, capital T. If you use T to represent a tall gene, it can be capital T, capital T. That means homozygous. Or it may not be homozygous. It may not be identical. It may be heterozygous. In that case, capital T, small t. So this pair of genes, which may be homozygous or heterozygous, that controls a given character is called what? An allele. Now, when they are in pairs, this pair, like capital T, is now in pair. This pair is called allelemorphs, but each of these pair is called an allele. So capital T, one capital T is an allele, the second capital T is an allele. The two capital T's are allelemorphs. These allelemorphs may be what? Homozygous, or they may be what? Heterozygous. Supposing you have capital T small t. This capital T small t is a pair now. It is called allelemorphs. But each of them, like capital T is an allele, small t is an allele. You may have capital B for maybe black coloration, or capital B small b. If it is capital B, that's a homozygous condition. And this homozygous condition is now called allelemorphs. It can be capital T, capital B small t, small b. That's a heterozygous condition that controls color. So this heterozygous condition, capital B small b, is also called allelemorphs. But each of them, like capital B is an allele, small b is an allele. Now, when we come to Mendel's first law of inheritance, you now know that what we call law of segregation of genes, or what we call law of separation of genes. You now see that two allelic genes can never enter one gamete. That during the formation of gametes, these allelic genes separate. We are going to get to that when we come to Mendelian laws. But we are still looking at terms in genetics. So we have seen allele, we have seen allelomorphs. Let's look at what we call genotype. What is a genotype? This is the genetic constitution of an individual. The genetic makeup of an individual is called what? The genotype. So your genotype may be AA, your genotype may be AS, your genotype may be what do you call it? SS. These are genotypes. A pair of gene, the genetic constitution of an individual is what? The genotype. Sometimes it may not be possible for you to look at the face and tell the genotype of an individual. For example, if somebody is AA or AS, it's not possible for you to just look at the person and say, this is AA. Or look at the person and say, this is AS. Sometimes it's difficult. But somehow, it can also, if somebody is SS, looking at the morphological appearance, physical appearance of an individual, you may have certain traits that may begin to tell you that this person is SS. So that is genotype, as you say, is the genetic constitution of an individual. What is phenotype also? The phenotype is the physical appearance of an individual. Where physical appearance is very simple, you can know a tall man, you can know a black man, you can know a white man, you can know a huge man, you can know a thin man, you can know a short man. This is the physical appearance of an individual. Somebody can be fair, somebody can be black. So this is the physical appearance of an individual. So phenotype is different from genotype. Why genotype tells of the genetic constitution, which cannot be determined by just looking at an individual. Phenotype, in a way, can tell you whether somebody is tall or whether somebody is short, whether somebody is slim or whether somebody is what, has weight, or whether somebody is a black man or whether somebody is a white man, or whether somebody is a tall man or whether somebody is a short man. So genotype has to key this. Now, let's look at what we call dominant character. Dominant character is a character that dominates. You also have recessive character. A recessive character is a character that is dominated. So if you are a tall man now, the gene for tallness has dominated. If you are a short man, maybe the gene for shortness has dominated. If you are a black person, maybe you are very black. The gene of blackness has dominated over the gene of fairness. So if you are very huge, the gene for huge weight has dominated over gene for thin. So that is what we call dominant character. For example, if your wife is a tall woman and you are a short man, and your children, all of them, or some of your children become tall, then for those tall children, we now know that the tall gene has dominated. This is what we call the dominant character. Then those genes that are dominated are called what? Recessive character. So you can have dominant character and recessive character. Now, I want to tell us, I want to look at what we call filial generation. Filial generation means the offspring of an individual. If you have married and you have children, those your children are first filial generation. These are your offspring that comes out from you. So filial generation are the offspring of an individual. So if you have children, your children are what? Your offspring. So that means first filial generation. Now, supposing your children have married and have other children, have their own children. Those your children are the what? Second filial generation. Your grandchildren are what? Second filial generation. Supposing your children now got also married and have children, then you now have children, your grandchildren. These are the third filial generation. So what are we talking? We are talking of filial generation. The filial generation are the offspring of an individual, which can run into first filial generation, second filial generation, third filial generation, fourth filial generation, fifth filial generation. And if I might ask you, do you know your fifth filial generation? Do you know even your fourth filial generation? But they exist. This is one of the wonders of the world. And so when we talk of evolution, people will begin to query, does it exist? But I want to tell you, since you know your grandchildren and you know your grandchildren, Some people know their grandchildren. That's how also your grandparents also there. So evolution is a reality. But then when we come to that bridge, we cross it. We are not discussing evolution yet. So, but we are thinking of filial generation to show us that you have first filial generation, second filial generation, third filial generation, fourth filial generation, fifth, even sixth to the 10th, to the 5th, 20th, to the 30th filial generation. And those of us that are claiming that we came from a particular village, you came from a particular town, who tell you? You may have migrated from a very far, where you have 5th or 10th filial generation have existed. So you find, when you trace your 10th filial generation, you may find out that you may have come from Cameroon. You may have even come from Chad, if care is not taken. So this is what we call filial generation. Now, close to this is what we call homologous chromosome. We have discussed about chromosome, something we call homologous chromosome. When identical chromosomes come together and stay, that is what we call homologous chromosome. We are going to come into that when we go to real Mendelian genetics. Of course, we saw this. I don't know. We did cell division, mitosis and meiosis. Yes, during the metaphase 2, metaphase 1 of meiotic division, we say that homologous chromosomes pay up at the equator of the cell. Yes, we can recall that. So we are now seeing that homologous is coming into us in genetics also. Now, we talk of locus, or what we call loci. I said that locus is the position of gene in a chromosome. The position occupied by a gene in a chromosome is called what? Locus. And then we have also discussed about homozygous condition and heterozygous condition. Homozygous condition is when the what? When the allele pair of genes that controls a given character are identical, you say homozygous. When they are not identical, you say it is heterozygous. Then we look at what we call a genome. A genome is a set of chromosomes corresponding to the haploid set of species. We look at that when we come into that. So we have seen that. We are looking at the somatic number in some organisms. So for example, in man, the somatic number, that is the number of chromosomes in the body cell, is 46. So the haploid number is 23. In Drosophila melanogaster, the somatic number of chromosomes in the body cell is 8, only 8. But our own is 46. So if you have 8 as the somatic number of chromosomes, 2n, what is the number in the haploid chromosome? The number is 4. That means n. 2n is equals to 8. N is equals to 4. For our own, 2n is 46 for human. What is n? That is the number in the haploid. Then n is equal to 23. That's why we have 23 number of chromosomes in our haploid cells, but 46 in our diploid. Now, look at another thing. Cattle. A cattle has 60 number of chromosomes in the body cells. So diploid number is 60. So 2n is equals to 60. How many number of chromosomes are found in the what? In the sex cell of a cattle? If 2n is equals to 60, n is equals to what? 30. So the haploid number in the sex cell is what? 30. In what? In a cattle. Now let us look at a horse. Equus caballus, that means horse. The number of chromosomes in the diploid cells of a horse is 46. The number of chromosomes in the diploid cell of a horse is 46. Then I will ask you, what is the number of chromosomes in the haploid cell?
23.
So very good. So if 2n is equals to 64, then the number in the haploid, which is n, is equals to 32. Let's look at a mouse or a mice. A mice has 2n is equals to 40. That means the diploid cell contains 40 number of chromosomes. Now I want to ask you, what is the number in a sex sex cell or haploid number of chromosomes? 2n is equals to, very good, 20. Then let's look at Zea mays, corn, ordinary corn we eat. The diploid number of chromosomes is 2n is equals to 20. Then in the haploid number, what is the haploid number?
10.
10. Very good. Let's look at fungus. Neurospira casa. Neurospira casa. The diploid number is 14. What is the number in the haploid?
7.
Very good. Let's look at monkey. Monkey. Macaca rhesus. Monkey. The diploid number is 42. That means 2n is equals to 42. What is the haploid number?
21.
Very good. Let's look at tomato. Lecopescum esculentum. Tomato. The diploid number in tomato is 24. What is n?
12.
Very good. Thank you very much. Now, let's look at chromosome morphology. Chromosome morphology. The structure of chromosome, how it look like. So, we say that chromosome is a rod-like substance that contains the genes. A rod-like substance that contains the genes that transfer character from parents to offspring. For example, in human being, the total number of chromosomes is 46, which is diploid number. 2n is equals to 46. Now, let's look at the shape of this chromosome. Let's look at the shape of this chromosome. Okay, now we have different shapes of chromosome. Now, in a chromosome, as we did when we did cell division, I tell you that chromosome has a center, and then the daughter chromatis that it holds. That center is called the centromere, where the hands are the daughter chromatis. We mentioned that when we did the cell division. Now, this centromere now, the position of centromere can be used to classify a chromosome. Now, when the centromere is at the center of a chromosome, that means the distance of the centromere now comes and stays at the center, so that the distance of the chromatis are equal, the upper chromatis and the lower chromatis. For example, I can use this biro to demonstrate. This is a biro. Now, when we talk of chromatis, chromatis is this place I hold my hand. The distance from here to up and distance from here to here are equal. So when the centromere are equal at the center of the chromosome, then that chromosome is said to be what? Metacentric. Metacentric. I use this pen as a demonstration. This is the center. When the centromere is at this center, the distance from here to up and distance from here to here are equal. So when the centromere are at the center of a chromosome, then what do you describe it? You say that the chromosome is what? Metacentric. Meta means at the center. Now, a situation may come when the chromosome, instead of being at the center, can shift small, so that the one length is greater than the other one. This chromosome is said to be submetacentric. Submetacentric. The other one is metacentric when it is equal. Then the other one is submetacentric when one is larger. When one arm is larger. Are you following me? So you have metacentric and what? Submetacentric chromosomes. Now, a time will now come when the centromere just moves almost at the top. Almost at the top of the chromosome, like this is a chromosome. The centromere, instead of staying at the center, or even staying here that is nearer one end, has come almost at the top here. That chromosome is said to be acrocentric chromosome. Acrocentric chromosome is almost at the top. Now, finally, a situation may arise when this chromosome will now stay at the top of one end. At the top of one end, come and stay at the top here. That chromosome is said to be telocentric because it is now at the top. The other chromatis is no more there. So that is what? Telocentric. Then sometimes the chromosome we have something like a knot that come and stay at the top. It's like a thread that stay at the top of the chromosome. Even when the centromere is at center, then there will be something like a knot or a thread at the top of one end. That is what we call satellite chromosome. Satellite chromosome. So we have described the morphological structure of chromosome. One, we say metacentric. When the centromere is 50-50, the distance is 50-50, is at the center. Then the other one is submetacentric. When the distance is not 50-50, but move towards one end, maybe 75 on one end and 25 on the other end. That is what we call submetacentric. Then it moves again, maybe to 90. The distance 90 at one end and 10 at the other end. Then you call it what? Acrocentric. Then it now comes 100%. Everything, the centromere is at the top. The other end, the other arm is no longer there. That is what we call telocentric. Then when you have a thread on top of the chromosome, then you call it what? Satellite chromosome. Satellite chromosome. These are the structures of a chromosome. You must know a chromosome as somebody that has passed through genetics. You must know what we call chromosome, and I thank God that you are doing it. So let's now go to Mendelian genetics. Mendelian genetics. It is very, very, very important. So we have already discussed mitosis and meiosis. We have discussed mitosis and meiosis. And then we have discussed the importance of mitosis. We also discussed the importance of meiosis. And I have given 10 differences between mitosis and meiosis. Thank you very much. We have looked at that. So now, we can now go, before we go into Mendelian genetics, that is the work of Mendel. Let's look at variation. Variation. What is variation? The difference in characteristics shown by organisms belonging to same natural population or species. Variations, differences exist, even in human beings. Some are tall, some are short, some are big, some are tiny. So these are the, some have big ears, some have small ears. Some have big heads, some have small heads. So these are the variations. But we have two types of variations. We have two types of variations. Variations, there is something we call continuous variation and discontinuous variation. Continuous variation and what we call discontinuous variation. Now, what is discontinuous variation? Discontinuous variation is something that one person has, the other person doesn't have. For example, rolling the tongue. There's a clear difference. I can roll my tongue. Please, can you roll your own tongue? Let me see. Rolling the tongue is what? Discontinuous variation. I can roll my tongue as I've rolled. Can you roll? Let me see your own. Oh, yeah. No way. Tereya Tokuma, you can. Solomon, you cannot. You cannot. Uma, can I see? Can you roll your tongue? Or Ayog? Ayog, roll your own tongue. Efe, roll your tongue. If you're able to roll your tongue, then you are with me. If you are not able, like Solomon cannot, then that's a discontinuous variation. I hope you understand. So rolling the tongue is a discontinuous variation.
What of twisting the eye?
Can you twist? I cannot twist my eye. Some people can twist their eye.
I can twist my eye.
Yes, that is a discontinuous. Because you can do it, but somebody else cannot do it. Some people can move their ear. Some people can, this guy can, I can see you twisting your eye. Me, I cannot do that. Now, some people can.
How do you define it? The discontinuous variation.
Very good. Yes, we are not. We will enter continuous now after we must have discussed discontinuous. Now, some people can shake their ear. They do like this. You see their ear shaking. Can you do that? I cannot do that. Some people cannot do that. So this is what we call discontinuous variation. Sickle cell anemia. It is also discontinuous. If somebody has sickle cell, another person will not have sickle cell. So these are discontinuous. So these are variations that are significant. So one person have it, another person, clear cut difference. Do you understand? There are many examples. Color blindness. Color blindness. Me, I can differentiate colors, but some people cannot differentiate colors. Even ability to taste with your tongue. I hope you understand. Some people cannot taste. Some people can taste. Smelling. Some people can smell. Some others cannot. So you have continuous variation. This is a what we call discontinuous variation. Are you following us? Now let us go into continuous variation. Continuous. Solomon,
Sir?
I don't know. You have your colleagues that are doing this course. How many? You may be 30 in a class. Then if you want to check the height of all of you, you spend more than 30 minutes or 1 hour because you see the tallest person, then you see another person that seems to be following the tallest, you see another person that be following, you see another person by following. Then you even get brackets and then you go down. So the variation is continuous as the number increases. Maybe you have 100 people, see the variation being continuous. This is what we call continuous variation. Are you following?
Yes, sir.
What of weight? You will see the fattest person. You see the person that follows it. You see the person that until you meet the person that is as tiny as so that is also continuous
variation.
Do you understand? Now,
yes, sir.
Color of the skin, fairness. You see, if somebody is black, you see the blackest person. You see somebody who is black as they following him. You see until you go continuous and you move to the one that is maybe considered fair. Then let us see fairness. You find somebody that look like Oyibo in this our class. So you see somebody that's fairer or following that person. Then you meet people like me that is fair. Then in the end, I think I'm fairer than Solomon. If I have not seen you to know whether I'm fairer than you. And then
you see me, sir. Don't worry, sir.
I'm fairer than you.
Yeah.
I'm fair somebody.
Okay. So that is what we call continuous what? Variation. The facial appearance, your face. If you are going to look at our faces, how they vary, you stay more than 3 hours trying to check our faces. That is also continuous what? Variation. Baldness. People that have bald head, you will continue to have some degrees of baldness. Some, their own have already disappeared. So some also have later. So you now continue to see this is what we call continuous what? Variation. Then we look at causes of variation. Causes of variation. Now, remember that during meiosis, during crossing over, something may happen. Accident can happen. That will result to new things happening. New organism, new person. Okay, even if we have what do you call them? People that are yellow but are not Oyibos in Nigeria. What do you call it? You see, their skin always get affected by sun.
We call those people albino.
Albino, so you now see it. So this is causes of variation. So it may be during crossing over that this thing happens. I hope you understand. Mutation can cause variation. Do you know the meaning of mutation? Mutation is a change. Mutation is a change in the arrangement of genes on the chromosome. And this mutation, what are the causes of mutation? People that can take hard drugs, X-ray. Yes. If you always go for X-ray, it can cause mutation. So that the children we are going to give birth, you see that something will just happen that they don't look like you again. Do you understand? So hard drugs,
yes, sir.
Smoking, what do you call it? X-rays, Then what radioactive radioactivity, where the Hiroshima, where they threw bomb, a lot of things changed. I hope you understand. So these are some of the things that can cause. Uma want to ask question. Solomon, what is your question?
No, sir, I don't have a question. I'm just
Okay. So we'll continue. Now, let's look at application of variation. How do we apply this variation? Variation can be applied in the following ways. Crime detection. Crime detection. If there is a crime, and then the police people can come there and take a picture, and then they will now call forensic people who will come there to take images and fingerprints. They can now use the fingerprint and begin to match it with people that are suspect, and you find out that in the end, the person who is a criminal will be detected. So it's used in crime detection. It also helps us in what? Blood transfusion. Before any person gives blood to you, that blood must be what? Checked. You scan the blood in the hospital to know whether it is what? Whether it is it can match. So variation helps us, because if it doesn't match and you keep do blood infusion transmission into the individual will begin to have problems. So compatibility is a very important something in the hospital in blood transfusion, and this thing helps us. It also helps us in determining the paternity of a child. For example, if a child is stolen and the police now got the person that has the child, and the child will now claim that it's his own or her own, it's a very simple matter. Just take the blood of that mother or father and take the blood of the child. Do the DNA isolation, do the PCR and polymerase chain reaction, and then do the what sequencing, and then do the blast and align them and do the blast. I tell you that if you do that, you find the original parent of that child, and that's settled the case. In the past, in the Bible, you remember Solomon was using wisdom to determine the paternity of a child in the Bible. But this time around, that knowledge cannot apply because many people are now intelligent. People have grown beyond that. So if you want to use Solomon's knowledge, I want to tell you that you fall like a pack of cards. So the new thing now, the modern technology is to use what? DNA technology. That is the modern molecular techniques to identify. So the variation helps us in determination of the paternity of a child. It also helps in adaptation and survival of an organism. So now let's look at transmittable characters and non-transmittable characters. Characters that can be transmitted from parents to the offspring, and characters that cannot be transmitted. Now, can you tell me those characters that you can inherit that can be transmitted from parents to the offspring? Can you try? Tell me. Characters that can be transmitted. Skin color, eye color, hair color, fingerprints, intelligence, height, tongue rolling, sickle cell anemia, badness, hemophilia.
Camera.
Yes. Some diseases.
Sir, blindness.
Are you sure? So if
Yes,
a mother
sir.
is blind, if a mother is blind, she will give birth to a blind child.
No,
Yes.
sir. Can't be possible.
It's not
No.
true. Some blindness are caused by environmental factors. Do you
Yes, sir.
understand? So, and some blindness
Okay,
are
sir,
caused
but
by
when I read on my
conjunctivitis.
own, sir, I saw blindness.
Eh?
When I read on my own, as in when I have some other tutorial class, you saw blindness.
No.
That is
So,
not true.
eye problem. You can talk of eye problem. I hope you understand.
Yes, sir.
Now, you can also have tallness is also a transmittable character. Do you understand? If you are a short man and you go and marry a short man, and then you think that you are a short man and go and marry a short woman, you want miracle to happen. Miracle will not happen.
You born the wife.
So the thing is that if you are short, marry a very tall woman. I hope you understand. Then the tendency is that you have mixed people that are tall and people that are short. The same thing with intelligence. So intelligence is a transmittable character. So if you decide to go and marry somebody who doesn't know pay, only that he has money, but talk of academics is zero, then I want to tell you that when you now have children, don't be surprised that some of them will not pass O-level. That is just it. So transmittable characters. But what are the characters that cannot be transmitted? What of armed robbery? Can it be transmitted?
No.
Is it a transmittable character? Armed robbery.
No, sir.
Is
No,
it a
sir.
transmittable character?
No, sir. No, sir.
When somebody picks your phone and refuses to give it to you, is it a transmittable character? Okay. What of lifespan? Lifespan. How long you stay in life? Is it a transmittable character?
No, sir.
It is. So some families are known to stay longer.
Are you sure?
Don't you know? Yes. Some families are known to stay long, to live longer. They reach 80 years. Why some have...
Me, I'm not in that family.
Eh?
Me, I'm not in that family. Our family will always reach 100.
So lifespan is transmittable. Do you understand? Some people are known to become the eldest people in the village, and it runs across their family. Do you understand? So lifespan is also transmittable character. Do you understand? Now, I want to tell us, some characters are controlled by even the environment and what? The parents. So, like intelligence. You may not be, you may not be very intelligent, but you are kept in a very good school. For example, if you are taken to Oxford Primary School now, the child is lifted from Nigeria and taken to Oxford. The environment is different. The environment will now begin to input on the child certain things that will make the child to be bright. You understand? So also, a child that is bright, but is not it doesn't have a very good background. So, but because that child is also inherited a very good gene, he will struggle and come out from that environment and pass very well. So you see that the development of an individual is a factor of two things: environment and inheritance. We call it nature-nurture controversy. Nature and nurture controversy. Nature is what we inherited. Nurture is the environment that you are coming from. Even some people are not thieves, but they stay in an environment where they are surrounded by what? People that are robbers. The tendency is that child will grow to become a robber. Do you understand? Somebody may inherit the gene for robbery, but he's put in a place that in fact everywhere is placed in a monk, and then the child is brought up in that kind of environment. The tendency to steal will reduce. So we call it nature-nurture controversy. And that's why some characters we don't know. That is still controversial, whether they are product of environment or product of what? Nature. Is that okay? So we now come to Mendelian genetics. Gregor Mendel, I told you, did mathematics and physics in his university days and graduated. But then he now had a call and then went into ministry, into monastery, and then joined them to become a priest. It was then that he started playing with these fruits, with red flowered plant and white flowered plant. In fact, the plant he played with is Pisum sativum. That is the plant Mendel played with. It's a garden pear called Pisum sativum. So he brought a tall flowered plant and a short flowered plant. He represented tall by capital T, capital T. And when you say capital T, it is a homozygous. Now, and then he now brought a dwarf plant called small t, and he crossbred them, only for him to find out that in the first layer generation, all the plants, all the Pisum sativum were all tall. Then he went to the flower, red flowered plants, Pisum sativum, called capital R, capital I. And then white flowered Pisum sativum, called small r, small r. He crossed both of them. And in the first flare generation, all of them came red. And that red is capital R, small r, capital R, small r. So that was what Mendel found out. Okay, that's where we call it a day today. So next week, we are going to go into Mendel's first law of inheritance and Mendel's second law of inheritance. And then we'll do some monohybrid cross, dihybrid cross, and trihybrid cross. We'll be able to finish this when we come next week. And we are already coming to the end of the facilitation. Only 7 people were able to join today out of 2,000 and something. What is the problem? So that is the problem. So, but all the same, we thank you for attending the lecture.
Sorry, sir. Excuse me, sir.
Yes.
Please, can you give me the definition for those? Listen, the first one you said was metacentric. I got that one.
Okay,
then
we are looking at the classification of chromosomes based on the position of the centromere. The first one is metacentric, when they are 50-50. The distance is 50-50. It is at the center. I hope you understand. But when you shift from the center and begin to move up again, so that maybe towards 75, the other one is now 25. It's nearer one side. That is submetacentric.
Submetacentric.
Yes. Then again, when it moves again towards one end, it is about the other end. The other is nearer the end. That is what we call acrocentric. Then you now have when acrocentric, A-C-R-O, acrocentric. The other one is metacentric, submetacentric. Then you now have acrocentric. Then when it has reached one end, no, the other arm is no longer there. That's what we call telocentric. Telocentric. Then a situation where we now have a kind of thread on top of one end of a chromosome. That is satellite chromosome. So I want to leave you here today, and next week we start. Thank you and God bless you. Bye. Bye.
Bye.